It's a key thing. Okay, that looks horrendous, doesn't it? This is the story that we're not going to read. We're going to look at in this picture. So start at the top left. And I did say this is a story of romance between one fine day, a plus strain and a minus strain. You see, poor old fungi, the Rhizopus doesn't even get to be male and female. They just get to be plus and minus compatible strains is what they call them. So, and they're compatible. If they're compatible, then they will grow towards each other. And so what they do is they start producing these swellings called progammetangia. Now, progammetangia are defined by swellings that start grow to each other from hyphae of compatible strains, and nuclei start to, you see the way the nuclei, this is what I meant, the nuclei are free-flowing in the hyphae, right? So the nuclei start to move into the progammetangia. Nuclei start to move into them, migrate into them. And then a cross wall sort of seals them off into the swellings. And then we call these things, then we call them gammetangia. So now there are two gammetangia and they touch. The barrier that separates them, dissolves. So they're trapped in there from these cross walls behind them, the nuclei are, and now they're allowed at each other, right? And they pair up, they fuse. It's fertilization. It's the fusion of gamete nuclei. And they form these zygotes, right? Just like plants, just like animals, this is fertilization. It's the fusion of gametes. They form these zygotes. But we call this collection of zygote nuclei, these diploid nuclei, we call these, the collection is held within a structure called a zygospore. So a young zygospore contains these newly formed zygote nuclei. And the zygospore then is a really resistant structure. So it's resistant to drying and to temperature to some extent. So even if the rest of the Rhizopus, suffers and dies, then this zygospore is built to survive. When conditions become favorable again, suitable, right, environmental conditions, then it will do this really weird thing, and you have to get this straight in your head. It germinates by meiosis. Now, it's really weird. Meiosis for us is for producing gametes. Okay, in plants, meiosis is not directly involved in producing gametes, but it's like a couple of stages before gametes are made. So strictly speaking, in plants, gametes are made by mitosis, but just a couple of steps before that, meiosis is involved in that process, right? Fungi flip this totally around and do this completely opposite kind of story. So they produce the gametes by mitosis. So those are just those nuclei were just regular nuclei that produced haploid nuclei produced by mitosis that were made a while ago, just happened to move into a progammetangium, got sealed off, and now pair up and fuse with each other. So they form diploid nuclei. Those diploid nuclei are short-lived. They're mostly diploid. They're mostly haploid. And their diploid structures, the zygospore thing, these zygote nuclei, that's a very short-lived, odd structure. Our odd, strange, short-lived, specialized structures are haploid, produced by meiosis. They use meiosis to germinate the odd thing, the diploid structure, and those meiosis, remember, produces haploid nuclei from diploid nuclei. That's what its job is. So it's not for producing gametes in this case. It produces a sporangium that germinates these, just like it's a sporangium, just like the sporangia we saw before, held on a sporangiophore, produces haploid spores by mitosis, and it's the same story as it was for the asexually reproductive structure from now on.